This lecture is over Chapter 5. We'll be talking about genetic mapping and genetic linkage in eukaryotes. So in Chapter 4, we talked about some of the extensions of Mendelian genetics, some of the things that cause us to deviate from Mendelian genetics. And today we're going to talk about deviations from Mendel's fourth postulate, namely independent assortment. So as we discussed last time, Mendel's laws of inheritance appear very clean, but in fact, for a large number of our traits, inheritance is a lot messier. There's a lot more things that go on. Examples of this that we talked about were epistasis, in which gene expression is dependent upon one or more modifier genes as to whether it's expressed or not. Or things like codominance, in which both genes get expressed. You have two dominant alleles. Or incomplete dominance, where there's a blending of the dominant alleles. And even aspects of expressivity and how we have a range of phenotype expression. It's not full expression. We can have gradations of that gene expression. All of those show that inheritance doesn't just necessarily follow the simple, clean rules and postulates as outlined by Mendel. Independent assortment states that as we look at two genes on two different homologous chromosomes, so here we have this gene A that's at this locus on this chromosome, and on a separate chromosome we have a locus that has this gene for B, and we can see that during meiosis, when we have independent assortment, we have four possible gametes that we can end up with, right? These are the possible gamete combinations that we could result in due to independent assortment. However, if these two loci are found on the same chromosome, then this alters the ability of those genes to independently assort. Because if they're on the same homologue, so on the same chromosome, and there's no exchange, we don't have any crossing over, then the only gametes that we'll end up with are A, big B, and little a, little b. Right? We call this linkage. Okay, so alleles are linked, or genes are linked when they're found on the same chromosome. Now, if we do have crossing over that occurs during meiosis between non-sister chromatids, then we can have genetic recombination. And it's possible if we have this, then what will result in is a chromosome that has no crossover. So we'd call this like a parental. And big A, big B, here's a parental, a, little b. And then we have two gametes in which we now have the crossover that occurred, in which we have a big A, little b, a, little b. And the reason this recombination occurs is because we form a chiasma, right? We have enzymes that come that then nick and basically cut these chromosomes apart. And then when we sometimes repaste them back together, we ligate the opposing or the non-sister chromatids together, and we get that genetic recombination. So a linkage ratio talks about whether there is complete linkage between two genes due to their close proximity. So the closer two alleles are to each other on a chromosome, so if we have A on one end, B on the other end, right? Chances are that these are far enough apart that the likelihood of a crossover event occurring between them is pretty high, and so they'll probably assort independently. However, if these two genes, if the two loci are very close together, the likelihood of a crossover occurring between them is reduced. And so we say the two alleles are completely linked or have complete linkage when they're very close together. And when this happens, that means we don't have recombination. We usually have very unique F2 phenotypic ratios. We can talk about linkage groups. Those are the genes on the same chromosome that are part of a linkage group, and the number of linkage groups, it should correspond to the haploid number of chromosomes. So in humans, we have 23 linkage groups. I already explained how we have crossing over that occurs at chiasmata, in which when we have those homologous chromosome pairs, we can synapse. They wrap around each other, and those X points where they have the intersection, that's where we have that genetic exchange. Something that we have identified that's unique about this is that the percentage of offspring that result from what we call a recombinant gametes, so the crossover, it depends on the distance between the two genes on the same chromosome. So the further the distance, the more recombinant gametes we'd expect to have. So, like I was saying, two genes located close to each other are less likely to have chiasma form between them. Now, this was determined by an early cell biologist named Struvant, and he compiled a bunch of data from crosses that he did in Drosophila, in fruit fly, and he started looking at what was the recombination frequency between genes that he knew were linked, and they were also additive. We'll talk about that a little bit later in the semester, what additive means. For right now, don't worry about it. But the frequency of this exchange, as he started getting these frequencies, he was able to estimate the relative distance. So he would take a female that was yellow and white with a male that obviously only had one. This was on the X chromosome. And he would do these combinations. And because these were fairly close to each other, most of the offspring of the females wouldn't have recombination, right? 99% of all of the offspring resulted in either wild type or yellow. Okay, but in the males, there was a few recombinants that would occur. Right, so here are females. You can see here's recombinant types. There are less than 5%, and so based off this, he was able to start saying, okay, how close are these two genes to each other? In a different gene, white and miniature, crossed with the wild type. Again, it was on the X chromosome. We found he had a lot higher amounts of recombinants. So here are the parental. They had the same phenotypes as the parentals, and these ones had different phenotypes than the parental strains. And here it was roughly 34.5%. So Struvant, he came up with this notion of like that percent recombination equals distance. And so he calculated what we now call map units. So 1% recombination between two genes on a chromosome is equal to 1 centimorgan or 1 map unit. Okay, these are relative distances. They're not exact ones. It's a relative distance that we utilize to map linkage groups. When we talk about crossover events, we can have two types. The first type is a single crossover. So a single crossover event occurs between two non-sister chromatids, and when we have this, typically what we see is that recombination gets observed in at least 50% of the gametes. And so this is an important number because 50% would result in what we call 50 map units apart. So as soon as two genes are more than 50 map units apart, crossing over can be expected to occur 100% of the time, which means that those two genes, for all intents and purposes, will independently assort. It's as if they're on different chromosomes if they're more than 50 map units apart. So if two chromosomes are 65 map units apart, recombination is going to occur 100% of the time. say, so here we can see if we had a chiasma forms here in a single crossover event, we would have no genetic recombination, but obviously if it occurs between them, we would have the recombination. And then when we look at these gametes, when we actually look at the chromosomes, again, just to name them, the two non-crossover chromosomes we would call the parentals. So this is a parental, right? No crossover occurred. This is a parental, no crossover occurred. And these are the recombinant chromosomes because we had a crossover event occur. Okay, that's a single crossover unit. These can be really useful in determining distances just between two linked genes. But what if we have, say, like 3 genes now? So now we're trying to find the order of these 3 genes. Well, a single crossover event isn't going to necessarily be able to help us. Now we're going to also need to use double crossover events, where we have a crossover that occurs here, and then, say, another chiasma forms between these genes, and we would go back. So now we could still have, in this case, the parental, right, the non-crossover gametes. And then the double crossover gametes would look like this. So starting here, we'd go A, we'd cross over, we'd be little b, and we cross back, and we'd be big C. Or little a, big B, cross back over, little c. Okay, we use double crossovers to determine distance between 3 linked genes. Now, a couple of tips when you're gonna be responsible for doing some genetic linkage problems. They can be very intimidating at first when you look at them, but there's some tips that'll get you through them really quick. First off, when we are presented with F2 data in which we have linked genes, if you look at the number of observed offspring, the highest number of observed offspring are always going to be your non-crossover gametes. So those are going to be your parentals. So this already tells me this was a parental strain and this was a parental strain. Okay, your double crossover phenotypes are going to be the smallest proportion. So the ones that have the smallest, the fewest individuals are going to be the double crossover event. Map distance, again, is that expected frequency of multiple exchanges between 2 genes predicted by the distance between them. So if they're further apart, that probability of undetected crossovers increases, right? And when we mean an undetected crossover, what we mean is when you have 2 genes that are very far apart, what can happen is that you can have a crossover event and then you could have a double crossover event. And we would never know that between these because it wouldn't be picked up by that, right? So, in order for us to calculate the map distance between 2 loci, what we can do is we can divide the total number of recombinant offspring by the total number of offspring. That gives us our percent recombination. And because map units equal 1%, well, each map unit equals 1% recombination, we can then get our map unit. If we can figure out what's the percent recombination between these, then we can get the map distance between them. And roughly, 1% recombination equals 1 map unit. That's about 1 million base pairs between those. It's about that. All right, let's do some problems. First, let's look at this cross that we have right here. We want to know, are A, B, and D linked? And if so, what is the gene order? Okay, so let's say we have 1,000 offspring. And out of those 1,000 offspring, these are what we have. Okay, now in a lot of these problems, sometimes your book, you've got, your book, or you've gotten very used to writing things out like up here. You might write this as this, right? Personally, when I do linkage problems, I like to draw my chromosomes. I find it much more much easier for me to do the problem. I'm less likely to make mistakes if I draw the chromosome. So I like to do it like this to remind myself what is the maternal chromosome and what is the paternal or the father's chromosome. Now, when you have it like this, the first one in each order, so the A would be the mom, the little a would be the dad, B would be the mom, little b would be the dad, D would be the mom, little d would be the dad. I'm randomly assigning mom and dad, but you keep that order. So here I'm drawing it to where I put one chromosome, this would be the mother's on top, the father's down below. Now in genetic problems, you're usually always going to be crossing it as a test cross. So with an individual that is homozygous recessive, so this is the cross that we've done, okay? And we now have 1,000 offspring. So first off, I want to know which ones are my parental offspring. So if I remember my tip number 1, that the highest number is always the parentals, then that should let me know right here, these are the parentals. Even if I didn't have this data up here, right, I can figure out which one are the parentals just because they're the largest number. And in fact, I can see that. Look, right, here's the parental, that big A, big B, big D. There's the other parental, A, B, D. I can see that right here. All of them are going to have this, right? There's the reason why we do linkage problems with a homozygous recessive is because no matter where we have chiasmas, right, we're only going to produce this gamete in all of the offspring. So that's why all of these problems that you'll do will always have the father or the other one of the parents will be homozygous recessive. Okay, now that second rule says the one that has the smallest number is going to be my double crossovers. So I would expect these guys to be the double crossovers. And again, I can see, probably should be able to see that in my data. Okay, so let's go through. Here's our parentals. These are recombinants, these are recombinants, these are recombinants. I can see the recombinants because, look, this one is recombinant. That one was a recombinant, right? Recombinant, recombinant. There's my recombinant. Now, when I start to find and calculate which ones are the recombinants, I want to pick gene pairs. So in this case, the recombinant right here is this position, right? That's the position in which recombination is occurring. It's occurring like this. Okay? For this one, the position of recombination is right here. Okay, and the only way that could happen is if I have this double crossover event. And for here, the position of recombination is right here, in which my recombination occurs there. So it's a test cross, again, because that male is the homozygous recessive. So now comes the question is, what's the gene order? Well, the way I figure that out is I need to figure out how many recombinants do I have between all of these gene pairs. And so we want to start, we're going to just randomly pick 2 gene pairs. First one, we're going to pick the AB. So again, this was my mother, or the female's gene order. Okay, so if I'm just taking between A and B, I'm not going to look, I'm basically going to not look at this section. I'm only going to look between A and B, and I'm going to see, was there recombination? Did recombination occur? So if I come to this one right here, I see big A, big B. Big A, big B, no crossover event. Little a, little b, no crossover event. I'm not looking again, I don't look at this position. I'm just looking between here. So these are not recombinants for AB. Over in these ones, though, big A, little b, that is recombination. Little a, big B, I had to have a recombination there. Same within this one. I had to have a recombination. So what I do is I take those recombinants, so 95 plus 47, that's 142. I'm going to divide them by the total number of offspring, which if I add all these up is 1,000. Okay, so divide by 1,000, that's going to give me a percent. Remember, I need to multiply by 100 to get, take it from a percent into, take my number to the percent so I can get my percent recombination. That gives me 14.2 centimorgans. Now I do the same thing for B and D. Okay, again, D, little d. In this case, I'm not going to be looking at this position. So I go through. In my first one, do I see a recombination between B and D? Yes, I'm big B and I'm little d. That was recombination, right? I had to cross over in order to get that gamete. Same thing there, right? So I know I'm going to add 5 down here. Do I do it here? Little b, little d. Nope, that's not a crossover. It's not a recombinant. Then I go to this one, big B, little d, or little b, big D. Yes, that only can occur if recombination. So I'm going to add 5 plus 95. That's 100. Divide by 1,000. Okay, times it by 100, right? That gives me my 10 centimorgans. Finally, I'll do the last thing for A and D. And the A and D one, this is the one that tends to be tricky when we do on the outside. Because again, at this point, you need to remember, we're not looking here. I don't care what's in this position. I'm just trying to say between A and D, was there a crossover event? I'm pretending like there's only 2 genes on these chromosomes. So as I look, I go here, big A, and then little d. Did a crossover have to occur for that to happen? Yes. Okay. I'll come over onto this one. Big A, little d. Did a crossover event have to occur for that one? Yes. So I'll add that. 5 plus 47. What about here? Big A, big D. Did crossover occur? No, they're on the same chromosome, so no crossover occurred. So here, I'm going to add my 5. I'm going to add my 47. That's 52 divided by 1,000 times 100 is 5.2 centimeters, or centimorgans. Okay, so now I have these distances. I could draw out my chromosome. And the way I would do it, draw my chromosome, I know that one is going to be, I'm going to take the one that's the largest, A and B. So I know those should be on my 2 ends, right? A and B are going to be my 2 ends, and those are roughly 14.2 centimorgans. A and D are the closest, so that's 5.2. B and D is the little bit further, that's 10. And if I added these up, so here's a question for you. Should I be alarmed that 10 plus 5.2 doesn't equal? So 10 plus 5.2 does not equal 14.2. Should I be concerned about that? No. Why? Because in our 2 chromosomes, actually this was the order. In our 2 chromosomes, it's possible when I have things that are further apart, so A and B being further apart, there's chances that I could have double crossover events in which I wouldn't see those crossovers in the in my gamete pool. If they occurred right here, I wouldn't know that I had a double crossover event between A and B. Even if it had occurred like this, to where I just had a crossover here and then a crossover here, I wouldn't know that I had a crossover. I would think that was a parental. And so, again, the further 2 genes are, the more likely you are to have a double crossover event and then obscure the number of recombinants. So these shouldn't really add up. The only time they would ever be perfect is when you're given a problem that says that there's no chromosomal interference. I just spoke about interference. So what is interference? Well, interference is inhibition of further crossover events. So when a chromosome forms a chiasma, sometimes that can interfere in the production of another chiasma close by. And it has to do with the kinetics and of the chromosomes being able to bend and form bonds with each other. And so there's a certain inhibition that occurs. So that can inhibit another crossover nearby, and that often can reduce the expected number of multiple crossovers. Okay, that would be interference. Now, that's not always the case. Sometimes you could actually have a crossover that when it occurs, it causes the DNA to coil on itself and it increases the chance. So we would have what we call negative interference, or we could have positive interference. Okay, so we can calculate interference, and we do that by calculating or quantifying the disparities between our interference. So we calculate what's called the coefficient of coincidence. We do this by looking at what the observed number of double crossover events are divided by the expected. And then once we get that, interference is really just 1 minus that coefficient. So when we have complete interference, that means you can never have a double crossover event between two genes. It means as soon as there's one chiasma, it's going to prevent any other chiasma from forming. When we have complete interference, this is when your numbers, the only time that your numbers between 3 genes will actually equal the same map distance as what you found between the 2 furthest genes. When we have positive interference, what that means is that we have fewer double crossover events than we would expect. So that I interference is a positive number. That means when we have a crossover, it makes it less likely it's positively interfering with the ability of another crossover to occur. Okay? So double crossover events are fewer than expected. When we have negative crossover, that means we have more double crossover events than expected. So now it's a negative interference, which basically means it's causing additional crossovers to occur. And our I would be a negative number. Okay, in general, when genes are very close together, we tend to have positive interference, right? When things are maybe further apart, then that's when we can have more negative. It should note that crossover doesn't just occur between non-sister chromatids. It also occurs between sister chromatids. So even during mitosis, we could have crossover. However, when we have crossover during mitosis, it doesn't create any new allelic combinations because sister chromatids have the exact same allele. So even though there's still chiasmas that form and we have genetic recombination, since all the alleles are the same, it doesn't mean anything. But we know what occurs. This is a picture of what we call harlequin chromosomes in which they've been radioactively labeled, the different nucleotides, and so we can see when there's crossover events have occurred. But they're sister chromatids, so they had all the same alleles to begin with. This information, we talk about it in Chapter 5. It was also a little bit talked about in Chapter 4, sex linkage. So on sex chromosomes, we don't have crossover. Okay, the X and the Y chromosome will not form chiasmas with each other because they're very, as you can see, they look very different. They really don't form, at least for most of the genes. There's some regions that are similar, and then that's what allows them to pair together. But we don't exchange genes. There's no way you can take a gene that's on the Y chromosome and exchange it with what's on the X chromosome. Therefore, we say that if there's an allele on a sex chromosome, we have sex linkage. So we co-inherit all the genes that are on the X chromosome and all the genes that are on the Y chromosome. Now, on the Y, we don't have that many chromosomes. It's not homologous with the X in the traditional sense. So it's not a big deal so much for the Y chromosome. But for the X chromosome, there's a lot of genes that are on that. And so since males only have a single copy of those genes, they're what we call hemizygous, only having one copy. And these genes, therefore, get inherited together. And so a male will have all of the genes that were found on their mother's X chromosomes. Sex linkage was first studied in Drosophila by a cell biologist by the name of Thomas Morgan. Again, he's the one that did that recombination and linkage. That's why we call them centimorgans. And what he discovered is that there was a mutation that would cause males to get white eyes. This is the first mutant gene that was ever really studied and talked about. And when he looked at these inheritance patterns, it was clearly related to the sex of the parent carrying that mutant allele. So when you look at F2 populations, he would find that 1/4 of the F2 males would have white eyes, but half of that would be 1/4 of all of the males when he did a cross between a female with red eyes and a male with white eyes. So in the first generation, none of them had white eyes, but then in the F2 generation, half of the males would have white eyes. So this is 1/4 of all, but half of all males would have white eyes. Likewise, when they did this reciprocal cross with females that had white eyes and males that had red eyes, what he found was that all males had white eyes, but none of the females did in the F1. And then when he looked at the F2 generations, he found that half of the females would have white eyes, and also half of the males would have white eyes. And so based off of these ratios, he basically came out and said, okay, look, there's something that is sex linked. We must have chromosomes that are different between males and females that don't have like this recombination that we've been talking about. And therefore, the males only have a single copy. So he already deduced that males have a single X, right, and that females have 2 X chromosomes based off of these ratios. And we can see that, right? So the reason why all the males have red eyes is because if this is a wild type trait, hence the plus on there, right? All of the males are going to get a dominant allele, a dominant wild type from the mom. If it's a recessive trait, okay, it's possible for the male to be recessive and have us and only have a negative, only a single W. And that's why the females in the first generation still have red eyes, because they have one of the X chromosomes has the wild type, the other X chromosome that they got from the father has the mutant. But it's a recessive mutant, so we still have it here. So this is just kind of showing it this way. We can look through these Punnett squares. So what I want you to take home from this is that whenever we find out that a trait is sex-linked, you need to change your Punnett squares a little bit. Because now you need to remember it's not just an autosome, it's an XY chromosome. So when I do a Punnett square, like these ones right here, what I tend to write, let's say we were doing this like a typical autosome. You might be tempted to go like this, right? Okay, so that's what the mother is. So the mother is wild type, dominant, and then the father is just little w. Okay, he doesn't have a chromosome here, so don't put anything there, right? That's what you want to make sure that you do. So sometimes I'll do this. just to remind myself. I'll do X's and I'll do Y's. Make sure it looks like a Y. I do Y. And then I'll use just the plus for the wild type, and for the mutant, I'll give it its number, so W. So this one is an XW, this is an X plus, and so we start going through. This would be X plus XW, X plus XW. This is going to be X plus Y, X plus Y. And now I can, because I've done this way where I've drawn in my Y's, I already can tell, okay, so here are my females. All of my females will be carriers, but they're still going to have red eyes. And all of my males are going to have the wild type allele from this cross. Okay, so make sure you do that with your Punnett squares. Write X's and Y. Don't just do letters like this, because it's going to get you confused. There's not an allele for this, right? It's a Y. That's for X-linked. So again, genes present on the X chromosome exhibit those unique patterns of inheritance, and it's because there's only one X chromosome in males and two in females. And it's really easy to identify sex linkage because our Y chromosome only has a couple genes. Most of the mutations are going to show up on the X, and when we have that, we'll be able to distinguish those in pedigrees, because we'll see an unequal proportion of males to females exhibiting the trait, regardless of whether it's dominant or recessive. In your Chapter 4 study guide, you had a couple questions asking you to identify whether you had traits that were autosomal dominant or X-linked dominant. Okay, a couple traits that we know that are on the X chromosome, genetic conditions, so red-green color blindness, hemophilia, where you don't form clotting factors. Those are just two examples in which we have that. Additionally, when you have an X-linked gene, females are the only ones that can be carriers, right? A male is either going to express it because they only have one chromosome. So they will always express it if they have that copy. But females can be a carrier in which they don't express it but still are carrying an X chromosome with them. The last two things we want to talk about with sex linkage, this really kind of corresponds to stuff that's found in Chapter 4. But sex-limited inheritance versus sex-influenced inheritance. So sex-limited occurs in cases where the expression of a specific phenotype is absolutely limited to one sex. Like you only see it in the one sex, it doesn't happen in the other sex. That's a sex-limited inherited trait. Sex-influenced is where the sex of an individual can influence the expression of a phenotype, but it's not limited to one sex or another. So let's look at a couple examples of this. One is feather plumage in chickens. Okay, this is an autosomal gene, so it's not on an X chromosome. And we have what we call hen feathering, in which you have the feather that look like a hen tail right here, and cock feathering, where you have the larger tail feathers and also the comb. Okay, cock feathering is controlled by a recessive allele, but it's only expressed in males. Therefore, this is a sex-limited trait. But we can see this, right? Hen feathering dominant in females. They're going to be hen feathered if we're homozygous. In males, they'll be hen feathered. Again, even in a male, it's still going to be hen feathered if they have a single copy of it. But if they're homozygous recessive, this is when we'll have cock feathering. But in the females, they're still not going to be cock feathered. They'll still appear hen feathered because there's hormones that influence this to actually occur. Male pattern baldness in humans, also autosomal gene. It behaves as dominant in males, but it's recessive in females. So it's kind of flipped, right? And we also know that it tends to be associated with how much testosterone is being produced in a male. So Here in females, you're only bald if you'd have, 2 copies of it, right? But in males, if you have a single copy of the gene, you'll develop baldness.